Hi, everyone. My name is Ibiayi Dagogo-Jack. I'm a thoracic oncologist at Mass General, and it's my pleasure to present today on BRAF alterations, or rather BRAF mutations in lung cancer. Okay, next slide. Perfect. These are my disclosures. Next slide. And so today in this short talk, I was hoping to discuss three main areas. First, we will describe the spectrum of BRAF alterations in non-small cell lung cancer. And yes, I kind of intentionally used the word alterations and not mutations. And next, we'll review how these diverse BRAF alterations can activate this key signaling cascade called the MAPK pathway. And finally, we're going to discuss the current strategies for targeting BRAF alterations, or rather BRAF mutations in this case in lung cancer. Next slide. And so when we think about activating oncogenic alterations in lung cancer, we think about mutations like EGFR mutations, rearrangements like ALK and ROS1, and then sometimes we can get copy number gain like amplifications in MET. BRAF can be altered or activated in a variety of ways in lung cancer, but by far and away, the most common way that BRAF is activated is through a mutation. In general, BRAF alterations are present in about 4 to 5% of non-small cell lung cancer across studies, and the most common way that BRAF is activated is mutations, and the predominant mutation is actually a particular substitution at the 600 position called BRAF V600E. There are other non-V600 BRAF mutations, and that account for about 1 to 2% of non-small cell lung cancers. And in addition to mutations, there are rare BRAF fusions or rearrangements. These can be present in the cancer at kind of original diagnosis, or they can emerge as an acquired resistance event, for example, in the context of receiving targeted therapy for EGFR. So captured here are images from this publication by the Foundation Medicine Group, really summarizing kind of the diversity of BRAF rearrangements or fusions that can be seen, as well as all the single point mutations that have been described in the BRAF gene or BRAF protein, kind of illustrating the point that the high peak right there, V600, is where the majority of these mutations are, but there are a clustering of other key mutations in the kinase domain. Next slide. And as mentioned, traditionally, we've really focused on V600 BRAF mutations, and that's because these were initially described in melanoma, where they're seen in about 40 to 50% of cutaneous melanomas, and we've borrowed a lot of our therapies from melanoma. In melanoma, it is rare to see non-V600 mutations, but in non-small cell lung cancer, we tend to think that, almost half or even more than half of mutations are non-V600 BRAF mutations. There have been kind of elegant studies over the last few years that have really taught us that it's more than just BRAF V600E and non-V600E, but rather when you look in the lab, there are functional differences between different BRAF mutations that have led us to actually subcategorize them, not just into V600 and non-V600, but rather class 1, 2, and 3. So class 1 is comprised kind of exclusively of that traditional V600 mutation, includes V600E and V600K. And then class 2 and class 3 really encompass the majority of the remaining mutations. And we'll talk about why we distinguish between class 2 and class 3 in upcoming slides. But as captured here, when you look across, and this is based on an internal study we did here at Mass General in collaboration with Dana-Farber, where we looked and kind of assigned BRAF mutations to distinct classes. And what we found is that there are still mutations that we don't know if they're class 1, 2, or 3, meaning we don't know the functional consequences of them. But for the most part, about 40 to 50% of mutations are class 1, and the remainder of the mutations, if we know their function, are kind of subcategorized into class 2 and class 3. And as you see here, if you look at different positions, whereas V600 predominates as a V600E, when you look at different positions like G469, there are different mutations that can arise. And that's important because they have different functional implications. So if we go to the next slide, it is important, another key concept that distinguishes BRAF mutations from when we think about MET, EGFR, and other things. When we think of MET and EGFR, these are kind of receptor tyrosine kinases or signal via this mechanism. They're on the surface of the membrane, they have an external signal, and they transmit it inside of the cell. BRAF, or the RAF proteins, are part of this complex and very important signaling cascade called the MAPK signaling cascade that has a bunch of important components, starting with that receptor tyrosine kinase on the surface. Then we have RAS proteins, the most important of which, or the one you're probably most familiar with, is KRAS. Then we have the RAF proteins. There's ARAF, BRAF, and CRAF. We're focusing on BRAF for this talk, but as you'll learn as we go on to a few more slides, CRAF matters too when we think about BRAF mutations. And downstream, we have MEK and ERK. Those are the key proteins that BRAF is signaling through to send proliferative signals to the cell. So the next slide. And so these proteins matter, and this signaling cascade matters when we think about this class 1, 2, and 3 BRAF mutations. As mentioned, class 1 is our classic V600 BRAF mutations. These, unlike in normal cells and in other BRAF mutations, where you need two BRAF proteins to join together and form a complex, we call that a dimer, to send signals to MEK and ERK, the V600 mutation in and of itself is fine. And so the protein can signal as a single, what we call a monomer. It doesn't need upstream input from the receptor tyrosine kinase or RAS, and it can activate MEK and ERK. Class 2 BRAF mutations are a little bit different. They too don't need RAS signaling or RTK signaling, but they actually rely on the dimer that we talked about. They need two BRAF proteins to trigger downstream signaling of MEK and ERK. Class 3 is a bit of an anomaly in that they are not truly activating mutations, in that they still need that receptor tyrosine kinase input and RAS input, but they basically amplify the signal from those inputs to signal for MEK and ERK. I know this is a lot of biology, but I'm going into it because I think it illustrates kind of the underlying principles why some medicines work for certain classes, but not others.